So I'm going to talk about interpreting the evolution of SARS coronavirus 2. So just to take a step back, I'm an evolutionary biologist, and I think it's important to remember that for most of the history of evolutionary biology, people didn't even know about genes or genotypes or anything like that. Here's Ernst Haeckel's Pedigree of Man, and you can see he's arranged all these creatures in this tree-like structure just based on looking at their phenotypes. And then, so it was only about 55 years ago that it became possible to also study evolution in terms of changes in genotype. So one of the first studies of this type was done by Emil Zuckerkandl and the famous chemist Linus Pauling. And what they did is they took the sequences of a bunch of myoglobin proteins that they were able to obtain, and they sort of compared those sequences. And although we're all very used to this type of analysis now, at the time it was actually heavily criticized by famous evolutionary biologists like Ernst Mayr or George Gaylord Simpson as sort of being trivial, because you couldn't learn anything by just looking at these mutations to these sequences. And indeed, Zuckerkandl and Pauling noted when they did this that it was not obvious how changes in these protein genotypes affected their phenotypes. They said there's no reason to expect that the extent of functional change in a polypeptide chain is proportional to the number of amino acid substitutions. It is the type rather than the number of substitutions that is decisive. So now we're 55 years later from that paper, and we've now gotten really good at characterizing the evolution of genotype at high resolution. In fact, we've gotten so good that most people forget that we ever used to study evolution any other way. So for instance, as I'm sure you all know, you can go to these databases and download hundreds of thousands of SARS-CoV-2 sequences right now. However, we often still want to know this old question of how these changes in genotype affect phenotype. So here's an article on this topic. It's asking, the coronavirus is mutating. What does that mean for us? And I hope what you can appreciate is the New York Times is just asking this same question that Zuckerkandl and Pauling were writing about 55 years ago, which is that it's not obvious to sort of just look at the number or the identity of amino acid substitutions in a protein and figure out the type of functional change that they cause. And so in my lab, we're really interested in studying how changes in genotype affect the phenotypes of viral proteins. So we're interested in this general problem of if you take some viral protein and you make some changes in the genotype, so some mutations, how are those going to affect the biochemical phenotypes that we think are relevant for the actual virus's fitness in nature? And so I'm going to talk about studying this mapping in the context of a specific protein from SARS-CoV-2. So the outline of my talk is, first I'm going to give a very brief introduction to the SARS-CoV-2 spike and its receptor binding domain. Then I'm going to take a step back and ask, how do coronavirus spikes more generally evolve with respect to immunity? Then I'm going to discuss how we can map how mutations affect the function of the RBD and how mutations affect the recognition of the RBD by monoclonal antibodies and polyclonal antibody immunity. So first, just a bit of brief background. As I'm sure you all know, on the surface of SARS-CoV-2 virions are these spike proteins. Here's one of these shown in red. And an important part of the spike is the receptor binding domain, which is an autonomously folding subdomain of the spike. And the function of the receptor binding domain, or the RBD, is to bind with high affinity to ACE2, which is the virus's receptor on the surface of target cells. We also know that neutralizing antibodies to the virus target the spike, and in many cases, they bind to the RBD and often act simply by blocking the interaction of the RBD with ACE2. And so to just underscore the importance of those antibodies, here's some experiments we recently completed with convalescent human sera. So each row in this plot is serum, as a different serum sample collected from different individuals at 2 different time points. And on the x-axis here is the neutralization titer, which ranges from, here's 1 to 100, here's 1 to 10,000. And all of the closed circles are the neutralization titers of these different individuals. So as is typically observed in convalescent sera, these neutralization titers mostly range from 1 to 100 to 1 to 10,000. And then what we did is we took these sera and we depleted all of the antibodies which bind to the RBD. And this is a small fraction of all of the antibodies against spike in the sera, but you can see when you get rid of those RBD targeting antibodies, In most cases, you dramatically decrease the neutralization titer. So we're going to the open circles now. And in fact, for many individuals, greater than 90%, in some cases as much as 99% of the neutralizing activity of the polyclonal antibodies is derived from anti-RBD antibodies. That's not always the case. There's a few individuals like this one right here where RBD targeting antibodies constitute less than half of the neutralizing activity, but in general, these are the most important component of the sera for neutralizing activity. Okay, so I've sort of shown you how this receptor binding domain is important for immune recognition. And now before I get into SARS-CoV-2 specifically, I want to take a step back and say, what do we know about how coronavirus spikes evolve? So really importantly, although all viruses evolve in sequence, only sometimes does that evolution in sequence change the antigenic phenotype. So two canonical examples among respiratory RNA viruses are measles virus and influenza virus. So these are both small RNA viruses with high mutation rates, but measles does not evolve to escape immunity. So in the old days, people were infected with this virus once in their lives, and then we have a vaccine that was developed in the 1960s, and it still works great today. At the other extreme is influenza virus, which evolves to escape immunity. So people are infected about every 5 years, and the vaccine needs to be updated annually to keep pace with this evolution. So we wanted to ask, where are coronavirus spikes going to fall on this spectrum? And so the way that we did this study is as follows. We looked at a coronavirus 229E, that's been circulating in humans for at least many decades. And what we did is we reconstructed viruses with the spikes of this coronavirus 229E from different time frames, 1984, 1992, 2001, 2008, and 2016. And then we took old human sera, so sera from 1985, which means this person was probably exposed to a virus like this one that circulated in 1984, and measured the neutralizing titer. So you can see this serum collected in 1985 has good neutralizing activity against the virus from 1984. And then we looked to see how that neutralizing activity was eroded over time by viral evolution. And you can see that as the virus continued to evolve, the neutralizing activity of the sera was eroded. So this is showing that coronavirus 229E undergoes antigenic evolution, more like influenza virus than measles virus. And in general, we did this experiment for lots of different human sera samples. And so each of these comes from a different individual, and the sera was collected at a different time frame, and the individuals are of different ages. That's all indicated at the top of the plot. And in each case, I've shaded in blue the period of time that the individual was alive prior to the collection of the serum. So here's the individual I just showed you. Here's serum that was collected in 1995. It neutralizes the older coronavirus 229E, but not the newer one. Here's sera from an adult in 2020. It neutralizes the full historical span. And then here is sera from a child collected in 2020. And you can see that it neutralizes only recent coronavirus 229E. So this is showing that as the virus evolves, it's changing its recognition by antibody immunity. One really important point that I want to bring up, because it's going to relate back to what I talked about for SARS-CoV-2, is this viral evolution, although it always erodes antibody immunity, it erodes the antibody immunity of different people at different rates. So here's serum from 3 different people, all collected, sometime around the late 1980s. And you can see that this individual, in just a few years of virus evolution, the neutralizing activity of their serum antibodies is completely obliterated by the virus's evolution. The titers are undetectable by 1992. In this case, there's sort of gradual erosion of the neutralizing activity. And then you can see this individual on the far right has what I would call evolution-resistant antibody immunity. And that you can see as the virus evolves forward in time, the neutralizing activity is slowly eroded, but it's eroded pretty gradually, so that even several decades later, there's still detectable neutralizing activity. And one of the things that we're really interested in doing is understanding the molecular bases of these differences among individuals. Because you can imagine if you were to create a vaccine against a coronavirus, you might like it to elicit immunity much more like this on the right, which is sort of more evolutionary resistant, than this immunity on the left, which is very sensitive to viral evolution. But at this point, I can't tell you the answer of what's different about the specificity of these antibodies. And then the last thing I'd just like to point out is that when you look at the evolution of coronavirus 229E, you see that most mutations are in the receptor binding domain. So for this virus, the receptor is aminopeptidase N, particularly in the loops that contact that receptor, and then to a lesser extent in the N-terminal domain. So again, at the top is the domain structure, and at the bottom is the variability at each site in the spike. And so another point that I just hope you can take away from is that this really sort of mirrors what we have seen so far in SARS-CoV-2, where there's a lot of evolution, particularly at sites like N501 or K4 or E484 that are sort of in these receptor binding loops. And then there's also some variation at other positions in the spike, in particular the N-terminal domain. So I think that coronavirus 229E is likely to provide a pretty good model for what we might expect for SARS-CoV-2. Okay, so now I've told you about the spike in its receptor binding domain, and I've told you how seasonal coronavirus spikes certainly evolve over time. So the next question I want to ask is, can we sort of actualize this information for understanding the specific evolution of the SARS-CoV-2 RBD? So for the SARS-CoV-2 RBD, what we'd really like to do is prospectively map how each mutation affects the relevant phenotypes. So importantly, it's impossible to measure true viral fitness in the lab. So we've focused on 3 biochemical phenotypes of the RBD that we think contribute to fitness. Those are, does the protein fold properly? Does it bind to ACE2 with high affinity? And can it be recognized by antiviral antibodies? And at an approximate level, we think of the evolution as occurring under the following constraints. The virus has to sort of keep its RBD good at folding and binding to ACE2, because those are absolutely essential for viral infectivity. And at the same time, it would sort of like to change the ability of the RBD to be recognized by antiviral antibodies as much as possible. So we think this more or less defines the biochemical constraints under which this evolution is occurring. So in order to map these biochemical phenotypes, we turned to a yeast display platform, which enables really high throughput experiments. So it turns out that you can just express the RBD on the surface of yeast. You can then detect how much properly folded RBD you have using a fluorescent tag. So in general, the protein won't be trafficked to the surface of the yeast if it's not properly folded. And you can also incubate these yeast with some fluorescently labeled molecule, such as ACE2, to see how well the RBD is able to bind to that molecule. And so you can sort of imagine using this, you can then run these yeast through flow cytometers and measure both the expression of properly folded RBD and binding to ACE2. And then importantly, we don't just have sort of one RBD on the yeast. We actually have a library of hundreds of thousands of different yeast, each expressing different RBD mutants. And in each yeast, we have a 16 nucleotide barcode, so we can rapidly deep sequence them and figure out which RBD mutant they have. And there's only about 4,000 single amino acid mutations for the RBD. So in fact, in our libraries, we have essentially all of the single amino acid mutants of the RBD, plus hundreds of thousands of double and triple mutants. But in this talk, I'm only going to talk about the single mutants. And so you can imagine just combining this library with the flow cytometer, you can pretty quickly measure the phenotypic effects of all of these mutations on these biochemical phenotypes. So just to show you what that looks like, here's the phenotypic maps for how mutations affect the function of the RBD. That's its ability to bind ACE2 and expression of properly folded protein. So each column in these heat maps is a different site, and then the color indicates the effect of that mutation. Red is bad for the biochemical phenotype, blue is good. So for instance, if we go right here, we can see that the mutation R355E is bad for ACE2 binding, and it's also bad for protein folding. And in general, you can see if you kind of scroll through these maps, there's a lot of red, so a lot of mutations are deleterious to these phenotypes. There's some white, so some mutations are neutral, and then there's scattered patches of blue. So some mutations are beneficial for those phenotypes. And this is super useful. Like we built these maps sort of over the summer of 2020. And as some of you may know, sort of later last year, people started to report a lot of viruses with a particular mutation called N501Y. So we could immediately go to the map and look up N501Y and see that that mutation increases the log 2 KD for ACE2 binding by 0.24 units. So it enhances ACE2 affinity, which could explain part of the reason it's been favored by selection. And you can also look at other mutations. You can see, for instance, E484K, which is another famous mutation, also very slightly increases ACE2 affinity, although the effect is a lot smaller than for N501Y. And you can look at other mutations that have sort of become famous, like K417N, and K417N right here actually decreases the ACE2 affinity a little bit, which may be why it's mostly found in conjunction with other mutations that enhance ACE2 affinity. So anyway, I think the useful thing about these maps is not only that we know the effects of those particular mutations, which have obviously now been measured by many other labs as well, but that every other mutation is in here. So I don't know what the next mutation that's going to come to high frequency is, but I'm sure there's going to be another one. And people can then just use this map to go see what the effect of that mutation is on these biochemical phenotypes. Okay, so now to return to my slides, we can also project this information on the RBD structure. I have a link here that does that, but in the interest of time, I'm not going to do that. Okay, so now I want to get to what I think is the last and sort of most important part of the talk, is what about how these mutations affect what I would say is the third important biochemical phenotype, which is how well antiviral antibodies are able to bind to the RBD. So I don't think it takes a lot of imagination to see how you can also use these libraries to measure that very easily. You just take your yeast, you incubate them with a fluorescent antibody, you run them through a flow cytometer to identify the yeast with RBD mutants that are binding less antibody than the rest of the population. And then you can deep sequence to see what are the mutations that are enriched in this antibody escape population. And I'm going to represent those data not in heat maps, but in these logo plots, where the height of a letter is proportional to sort of how well that mutation escapes antibody binding, specifically the fraction of all yeasts containing that mutation that fall in the antibody escape FACS bin. So the main thing you need to remember when I show these maps of antibody escape is if you see a tall letter, that means that mutation escapes binding by the antibody. Okay, so here is just an example, is an escape map for one particular monoclonal antibody called COV22499. And if you look at this map, I'm only showing the sites that are sort of important or interesting in some way. You can see that the antibody escape mutations occur at just a small number of sites, around the 443 to 450 loop, and then sort of in the high 490s on the receptor binding ridge. And this makes a little more sense if we project where these escape mutations occur onto the structure of the RBD. So here I'm now showing, let me see if I can refresh this to get the colors better. All right, I'm now showing the structure of the RBD. So that's here. The ACE2 is this larger helical thing at the top, and the RBD is this more beta sheet thing, rich thing here at the bottom. And then each of these, okay, now I was able to get the ACE2 in a dark color and the RBD in a light color. And then, as I've described to you, for each site in the RBD, we've measured how mutations affect antibody binding. So most sites, mutations have no effect. You can see there are a few sites like 447, 496, et cetera, where mutations have big effects on antibody binding. So we can go and we can look and we can see where those sites are on the RBD, and you can see they all cluster in this one location right here, which must be where the antibody binds. And so this not only maps out the antibody escape mutations, you can kind of think of it as a useful epitope mapping strategy. And maybe for a monoclonal antibody, the ability to do the epitope mapping isn't all that impressive, because you could do the same thing using structural biology means. But as I'll get to in a little bit, this technique can obviously be generalized to polyclonal serum antibodies, where it's much harder to determine epitopes using structural biology approaches. Okay, so that sort of shows what one of these escape maps look like. And then what we wanted to do is test if our measurements of these biochemical phenotypes could actually explain how the virus evolves. So to do this, we worked with collaborators who grew a spike-expressing VSV in the presence of various ones of these monoclonal antibodies to then see what escape mutations were observed. So here's an example of one of these experiments for the antibody I just described to you, CoV2-2499. They grew the virus in the presence of this antibody 16 times. 5 times they got escape mutations, and this is what the escape mutations were. So now I want to ask, can we go back to all of our measurements of these biochemical phenotypes and understand why the virus evolved the way that it did? So if we just look at the first biochemical phenotype, which is, does the mutation escape antibody binding? You can see that there's a lot of tall letters, so a lot of mutations that escape antibody binding, and the ones that actually emerge in the virus are among those escape mutations. However, the predictive power here is low because we can't say why we got these two mutations rather than all the other possible ones. But we can then add on other information we have. So first of all, I'm going to subset this map of the effects of all amino acid mutations to just those mutations that the virus can reach by changing a single nucleotide. So only some of the mutations are accessible by single nucleotide changes, including the ones that actually occur. And then importantly, I told you how the virus is also under constraint on other biochemical phenotypes. In particular, the RBD has to retain the ability to bind to ACE2. And we also measured how all mutations affect that phenotype. So here I've now colored all of these letters by their effect on ACE2 binding affinity. Brown means that retains strong ACE2 binding affinity, and yellow means the ACE2 affinity is really bad. And when you look, you can now see that there's only a handful of mutations that escape antibody binding, so have tall letters, while also retaining good ACE2 binding affinity, so being brown. And the ones that the virus actually gets are among that small subset of mutations. Another way to look at these same data is to project all of the mutations to the RBD onto this space of these two biochemical phenotypes. How well does the mutation escape antibody binding, and how well does the mutation retain ACE2 affinity? And teleologically speaking, you'd think that the virus would want to escape by getting mutations in the upper right of this space of biochemical phenotypes, because those are big escape mutations that don't incur a cost on ACE2 binding. And after I gray out all of the mutations that cannot be accessed by a single nucleotide change, you see there are only a handful of mutations that satisfy that constraint, and the ones the virus actually gets are among those. And we've done this for a variety of other antibodies, and consistently, the mutations the virus gets are as far in the upper right as is possible to get, given the constraints on the biochemical phenotypes. So I think this is cool. These phenotypic maps can really sort of explain to a substantial degree which mutations are selected under antibody pressure, in addition to just allowing us to look at mutations and tell if they're going to escape the antibody. And so we've now built these escape maps for lots of antibodies, including many of the lead clinical ones. I think the only one that's not in these right now is LY-CoV55, where we're hoping to post the preprint of that one today. But we have the Regeneron cocktail, a variety of other antibodies, the ones in the AstraZeneca cocktail, and one of the Eli Lilly antibodies here. And so you're welcome to go to these links and look up the escape maps if you sort of want to know how some mutation is going to affect binding by those antibodies. Okay, but although I think monoclonal antibodies have sort of been a useful stopgap, as I'm sure everyone knows, the thing that we really care most about is the polyclonal antibodies elicited by vaccination. And just to sort of emphasize the obvious, the difference is that a monoclonal antibody, as it left here, is a defined antibody that binds to one place on a virus. And usually it's possible to the virus to get a single mutation where that antibody binds that ablates the binding. In contrast, the polyclonal immune response can, at least in principle, make antibodies that bind to many different regions of the virus. And in that case, no single mutation is going to escape from all of them. Importantly, the polyclonal immune response isn't always effectively polyclonal. Sometimes it's highly focused. And the extent of focusing of polyclonal antibodies crucially shapes antigenic evolution. So for influenza virus, the human polyclonal antibody immunity is often highly focused, such that a single mutation can often reduce neutralization of influenza virus by greater than 10-fold with respect to polyclonal human serum. In contrast, for measles virus, the polyclonal antibody response is highly distributed across the surface proteins, targeting many epitopes with similar potency, such that usually no single mutation has a large effect. So we sort of applied the same mapping approach I showed you to sort of build these escape maps for convalescent human sera. And there's a lot of these maps that are described in this paper here. Here I'm just showing a couple of representative ones. And so first of all, you can see that the regions where mutations escape binding by the convalescent human sera fall in three main epitopes. One we're calling the receptor binding ridge, one the 443 to 450 loop, which would correspond to class 3 antibodies in the Pamela Bjorkman classification. And then there's also this sort of epitope in the RBD core. And you can see, so those are the main epitopes. In particular, we think the main neutralizing epitopes are the receptor binding ridge in the 443 to 450 loop. And you can see that different individuals have, sorry, I'm gonna go back here, I guess first. You can see that different individuals have immunity that's targeting different ones of these epitopes more or less. So for instance, this person's immunity is highly focused on the epitope centered around E484. This person's immunity is more focused on the 443 to 450 loop. But if you sort of average across all of the different individuals we've looked at, which includes a lot more than the 4 I'm showing here, the most important site is generally E484, which, as you know, is mutated in a variety of viral lineages. So it looks like sort of the worst single antigenic mutation is already out there in the virus. And as I'm sure David Ho will talk about, people are looking into its antigenic impact. But as for coronavirus 229E, people are affected differently by different viral mutations. So for instance, if you look down here for subject K, no single mutation to the RBD has a large effect on antibody binding. So we might speculate that this individual will have relatively evolution-resistant antibody immunity, and they may be able to stand up to a large amount of viral evolution.
Sort of near the end,
okay.
Jesse. Yeah,
okay, sounds good. And then finally, I just want to point out these differences between people validated neutralization assays. So here's just an example. As the map suggests, subject C has their neutralizing activity almost greatly reduced by 2 orders of magnitude by E484K. For subject G, that has no effect, but as the map suggests, mutations in the 443 to 450 loop, like G446V, have big effects. Okay, so just to kind of conclude, relating changes in genotype to phenotype is obviously an old challenge in evolutionary biology. I talked about how Zucker, Candle, and Pauling were worrying about it 55 years ago, and now we're worrying about it in the context of coronaviruses. But what I think is new is we have so many genotypes that really functionalizing or interpreting the effects of these mutations has become the scientific bottleneck. And so I've kind of described to you what we consider some new approaches that can really map the functional effects of mutations at a scale that's commensurate at the rate that we're generating genotypic data. Okay, and finally, I'd just like to thank the people in my lab who did this work, in particular, Tyler Starr and Ali Greeny, who are a postdoc and grad student who really led all of the mapping work, as well as Rachel, Sarah, Kate, and a number of wonderful collaborators. Thanks.
All right, Jesse, fabulous talk. Thanks so much. This is all very, very important and really amazing. So I had a couple of questions I could start off with. So one of the important things that you brought up in the middle was the durability of the response, and those people with the other coronavirus antibody responses were very variably stable or not stable over a long time. So to what extent do you think that's correlating with the specific antibody they made versus some aspect of the lifetime of their B cell memory that's just individual?
Yes, so I think it's, I think we have to distinguish 2 points, one of which is viral evolution and one of which is durability of immunity. So importantly, in all of these data I showed here, we're just looking at the same serum sample. So here it's the serum sample collected in 1985, and we're measuring that against all the different viruses. So how durable the antibodies or the B cells are is a separate question. But what these plots are showing is even if those antibodies or B cells are durable, the virus is eroding the effectiveness of the antibodies. So I think clearly the difference has to be that the antibodies from this person are targeting different epitopes, and in particular epitopes that are changing less during the evolution of the coronavirus, and the antibodies of this person are targeting epitopes that are changing very rapidly during the evolution of the coronavirus. Like I said, we haven't worked out exactly what epitopes those are, although as I'm sure you're aware, most of the neutralizing activity, at least for SARS-CoV-2, and although it's not worked out yet for coronavirus 229E, probably coronavirus 229E target the RBD or the NTD. So whether the differences can be explained at domain level, subdomain level targeting like that, or whether it really comes down to specific epitopes in the NTD or RBD, I can't give an answer to that yet.
Right. So the durability in this setting will really depend on whether the virus has the tendency to be able to mutate away or not from that particular response.
Exactly, exactly.
Cool. All right, so let me open up to questions. Let's see, we've got some question. Okay, so I'll just read. This is from Mariska Van Huysen. So the question is, I was wondering whether it's possible that a combination of mutations is beneficial where the individual mutations are deleterious. One could imagine that a combination of mutations would stabilize the RBD in the case that a single mutation is destabilizing. So of course we think about compensatory mutations, right?
Yeah, I think that's a great question. So at this point, we don't have systematic information on epistasis. We have some multiple mutants in the library, and I think this question will take on growing importance as time goes on. I mean, early in the pandemic, you could essentially think of most of the spike mutants as single mutants around some central sequence, and already that's starting to be less true. So there's certainly a simple form of compensation, which I think our data capture well, which is I think we're probably seeing, for instance, in the 501Y.V2 lineage, it has a mutation K417N, which decreases ACE2 affinity, and a mutation N501Y that increases ACE2 affinity, and they just sort of cancel out on the scale of ACE2 affinity. So that's pretty simple to understand. But certainly, as you point out, a lot of these mutations are also clustered in the three-dimensional structure. So you could also have the potential for really complex conformational epistasis depending on, you know, how the side chains are packing or something like that. And yeah, I think that's an important, interesting question. We're trying to work on it. At this point, I don't have anything intelligent to say about it other than it's an important question.
Right. In the HIV field, we're very used to that in the context of resistance to drugs, where you've worked on a protease gene will first evolve resistance to a drug and then be sick and have to compensate with another mutation that doesn't change the drug resistance, but makes it back to normal function.
Yeah, and I would point out that in sort of acute respiratory viruses that usually cause self-limiting infections, I think compensatory mutations happen much less than they do in a chronic infection like HIV. And instead, you get the same endpoint, but you tend to get it by permissive mutations, which is namely what you would call the compensatory mutation happens first, and then the second mutation happens on that background. Because of the nature of the rapid transmission of respiratory viruses, they have a harder time handling, you know, sort of being in a transient low fitness state. But you get to the same endpoint.
Right. So now one from Molly Brzezowski, that what do we understand about population genetic parameters of previous coronaviruses and how they compare numbers of viruses within and across individuals?
Yeah, that's a great question, Molly. So I actually had a nice phylogenetic tree here, which I took out in the interest of time. But so human seasonal coronaviruses, particularly coronavirus 229E, tend to have a ladder-like phylogenetic tree similar to influenza virus. And essentially, this is a characteristic of a population which is changing rapidly over time, but for which the standing diversity is low. And the time to most recent common ancestor is usually never more than a couple of years. So certainly those are the primary evolutionary dynamics, which I think in some ways makes, you know, respiratory viruses like flu and coronaviruses easier than something like HIV, where there's high standing diversity as well as change over time. You know, why exactly that's the evolutionary pattern that's seen in you know, some of these respiratory viruses is, there are theories, but it's harder to tell. As far as the you know, effective population sizes, the effective population sizes of these respiratory viruses is actually sort of surprisingly low given how large the census population sizes are. And the reason is because there's a lot of extremely narrow transmission bottlenecks. The transmission bottlenecks are probably a couple of infectious particles. So I think the reason that evolution honestly doesn't happen faster than it does is because the intrahost selection is constantly getting broken up by these narrow transmission bottlenecks.
Right. Every transmission event is a clonal event almost. Yeah.
Or at least near clonal, yeah.
So one more, Raul Rabadon asks, have you checked homoplasies in the phylogenetic tree, a sign of convergent evolution? You'll have to define homoplasies for us.
Yeah, so homoplasies is just this like complicated word that evolutionary biologists use to mean the same mutation happening multiple times. At least that's my understanding. Someone can correct me if I'm wrong. And we haven't specifically, but there's a lot of sort of recurrent mutations. So like this N501Y mutation has emerged 3 different times. So it's in the so-called the so-called UK lineage, the 501Y.V2 and 501Y.V3. So probably the 3 most major lineages. E484K has appeared multiple times. So there's definitely a lot of recurrent selection of the same mutations going on. It's actually, you expect some amount of recurrent of the same mutations when you have a high mutation rate and a small genome size, but I'm actually surprised how much it's happened. I mean, it's really extraordinary how many times some of these mutations have independently arisen.
Yeah, I mean, it's telling us, I guess, the pressure is the same, and it's interesting how monolithic the pressure is, I guess.
Yeah, it's very true.
So one more, we're running out of time, but John Hunt asked sort of a related question. So he asked you, do you buy the premise that multi-mutant viruses may be getting selected during persistent infections in immunocompromised patients? Right?
Yeah, so this has certainly been postulated that some of these viruses, these lineages that are expanding, have sort of more mutations than you would expect given the molecular clock. Although you have to add the caveat that, I mean, molecular clocks are not that accurate over very short time frames, particularly when you're looking at mutations that haven't necessarily gone to fixation. I think it's certainly plausible they've arisen during immunocompromised patient infections, but I don't know for sure. I mean, we do also expect that because of the nature of the selection pressure, variants with more mutations are going to be favored because they're the ones that get away from the immunity more. I mean, this is well known in flu that, even where hasn't been postulated the persistent infection thing, the trunk of the phylogenetic tree tends to have more mutations at certain sites. So I don't know. I think the persistent infection thing is definitely plausible, but at least as far as I can tell, it's not proven. That's for sure.
Yeah, I guess we don't know the settings where the selections are most happening, right?
Yeah.
So a last one, but we got to move on. But the last one is just a simple one, I think, from Kai-Yin. So your binding data is based on things expressed in yeast. What about differences with the yeast binding versus real virus?
Yeah, so as far as the ACE2 binding and the binding by monoclonal antibodies, I mean, we've done at this point tons of validations with pseudovirus and mammalian express protein and comparing to literature, and they validate extremely well. The main differences, I think, where you look at polyclonal serum antibodies, and the main difference is the RBD is monomeric. And so we see sort of more binding to the epitopes that are included in the full spike because those are sort of harder to get to in the full spike than they are in our system. So those would be what like Pamela Bjorkman and Christopher Barnes would call like the class 1 antibodies. I think our serum mapping overestimates the importance of class 1 antibodies compared to what they actually are in the context of full spike.